Hello, everybody. We are back to our fourth webinar of our Brainstorm webinar series. And in the past day, if you followed our webinars, we went through sample analysis, we went through sample preparation, how we can prepare cell sample, EV sample, how we can image them. But what we teased a little bit and what we didn't go so deep is actually the sample analysis and how we can extract meaning from data from this storm imaging. So today is going to be the last part of the webinar series and all focused on understanding clustering organization in CODI. And what is CODI? You will learn much more later on in the conversation. And so today, as the past days, it's going to be a podcast webinar style. So please feel free to pop in your question at any time during our conversation. And we will make sure to ask our expert today. And as always, I'm going to read out the general rules for today. So this webinar, you have a little navigation panel on the side where you have some handouts that you can access for your information. And importantly, you will find the chat functionality if you click on the expand icon. And today's webinar will last approximately 60 minutes. And as usual, it will be recorded so that it will be available for you on the website and for later referencing. And if you think it's useful, for sharing with any of your colleagues. And so we're going to start again by introducing ourselves right now. So my name is Laura, and I'm a field application scientist for ONI. So what this means is you may know me from installations of new systems, from trainings, from demo days, or from conferences. And today we have with us Corey. Corey, can you introduce yourself?
Sure. Thanks, Laura. So I'm Corey. I am the product manager for software here at ONI. So I'm responsible for our CODI platform, which kind of started out as our single molecule analysis platform, but is now expanding into acquisition and reporting and all this great stuff with our newly released AutoEV. Got a background in single molecule imaging. I did my PhD in this stuff. Been doing it for about 10 years now, and I'm really excited about what we're doing here at ONI that's combining the acquisition, making the sample preparation really easy, making the acquisition really easy, making the analysis really easy so that you can get a report that you can understand in a really quick amount of time. So that's what I'm doing here at ONI as a product manager person.
Thank you, Corey. And to get this conversation started, we actually gonna get a quick poll for you. So we are gonna ask you the first question, dear audience, and that is, what is your level of expertise right now when it comes to single molecule localization microscopy analysis? Maybe you have done zero image analysis, that's okay. You have done some maybe single molecule analysis, or you have done some but not single molecule analysis, which would be very interesting to compare. Or you are a pro and write your own code. Kudos for that. So what is your level of expertise right now in single molecule localization analysis? Gonna give it a few seconds and then reveal the answers from you. Okay, so we have a very, very broad mix of answers from you today. So we have some 0 to some experience of single molecule localization microscopy. That is very great. We're gonna go through a lot of analysis today. And then we have 17% of people that have done some image analysis, but not single molecule, and even some that are real pros. Fantastic to hear about this mixed audience today. It will be hopefully a very exciting discussion. And as a reminder, please leave us your questions in the chat and we will make sure to ask them to Corey. Corey, let's start with the basics. So We are going to assume that we have a really nice sample prepared and we are ready to image it. Could you guide us a little bit how this process would work from the imaging to maybe the final data output?
Sure. Okay, that's a good one. That's a good place to start, I think. It's a good place to kind of level set all the words too, because analysis can mean a lot of things, acquisition can mean a lot of things, even image can mean a lot of things. So if you go to the next slide here, I have a little workflow here of how we go from the acquisition of the data, which is the camera feed that you see on the left, all the way through maybe some clustering results that come out as data files that you can then analyze and plot in other software and be able to like show as a graph for your report or add to your publication or something like that. So the first step is generating your awesome camera images. This is what we call the acquisition, right? So you'll put your slide, if it's like an EV chip, you'll put your 4-lane assay chip onto the nano imager. You could use a tool like our Auto EV tool to automatically start your imaging. And you'll get this wonderful blinking images, and that's our raw data. That's our raw single molecule data, right? What we need to see here is really nice blinking kinetics, and we can do some analysis on these images themselves. You can do blinking rate, or what we call duty cycle analysis of the fluorophores. It's basically how long they're on and they're off. And you can analyze how bright or intense or how many photons are in each of the fluorophores that are emitting in each of the individual diffraction limited spots. On your nano imager, while your acquisition is happening, there's also this process that we call the localization process, where my colleagues have gone over this in the past few days, right? So I think you guys are probably familiar with this now, but we're taking each one of those individual dots that you see on the screen, and we're running it through a localization algorithm that gives us the center of each of those dots with about 10 times better precision than what we can get just from the diffraction limited image. So that's what gives us that super resolution, right? Is the ability to run those spots through our localization algorithms and get the center, the position of those emitters with, nanometer scale accuracy. What that looks like in terms of data is actually, it's not an image itself, it's actually just a big list, right? It's a big list of localizations and a whole bunch of properties that are associated with them. Some of those properties are their spatial position, so in X, Y, and even in Z sometimes if you want to do 3D imaging. And then a whole bunch of other properties like the size of the point stretch function, so that's what the individual dots are called, or the intensity of them, or the local background information. And all of those parameters, all those localization properties are really important to making sure that you have good quality data. Then that's what I would call the localization list, and I would call that the point cloud. You'll hear us refer to that in that way several times. The next step is what I refer to anyway as the single molecule analysis, right? So what are you doing to that point cloud data, that localization list, to turn it into quantitative and meaningful data for you that you can then put into your graph and compare across different conditions and experiments and stuff like that, right? And so one typical example of this is clustering your localizations into spatial clusters, so things that are spatially close to each other, organizing them so that you're able to detect things that are close together versus things that are far apart. We call that clustering, and there's different algorithms that I'll probably get into a little bit later about how we do that. The output of that is similar to the localization. It's a list of clusters this time, so it's like one level down, right? Where you have information about each of those clusters, the center of it, and other properties like how many localizations were in there, in each of the different channels if you're imaging in multicolor, the size of it, other important information that you can use to compare different experimental conditions. Then if you're doing this on multiple experiments, you can take all that clustering data, like I said, put it in something like Excel, draw some graphs, and you can put that into your reports or into your publications. So starting from the acquisition all the way through to your pretty reports. That's the general workflow here, and we'll dive into it a little bit, I think, in a little bit.
Yes, thank you for this overview. We have heard a lot of terms, but we're going to dive deeper into them. Don't you worry. And first question with you would be more related to the localization and properties. Can you briefly comment on what are we looking out for? What are the best properties localization should have to have really good accuracy and to that I can be confident this is real signal? How can I distinguish that?
It's a really, really good question. It's a really important one too. So the localization process is going to basically localize anything that it sees as an individual spot on the camera and add it to that localization list. And what we can do with those properties, other properties that I'll get into in a little bit, is eventually then in a second step, and I'll show you some of this later, sneak peek, is filter that so that what we're doing is just retaining the best data that gives us our real true single molecule signal. Because most of the blinking that we have is stochastic when we're using storm or random, it's really hard to control and ensure that every single localization is just one single fluorophore that's emitting in that diffraction limited spot. When it's not the case, you can end up having a localization error because you've got like two molecules on at the same time, and you're basically trying to average the center of them, and that gives you a little bit of an error. With the localization properties, so there's things like size of your point spread function and intensity of the localization, you can actually restrain those using some filtering that I'll show in a little bit to ensure that the data that you're actually going to continue down to analyze is only your really truly single molecule data, so that you're actually using the correct and good localization data and not things that are coming either from multiple fluorophores that are emitting in the same spatial localization at the same time, or just some background. Sometimes they're just kind of background signal floating in your sample, and you can remove that as well because it's usually dimmer or larger or something like that. And so, well, I'll show you later about some of the ways that you can get rid of some of those aberrant localizations, but filtering them by those different properties is really critical to ensuring that you're getting the best data.
Absolutely. And can you briefly comment, in your experience, how much does it take from my nice blinking image to this localization image? So in other words, how much does it take for the software to process these blinks and display me as localizations?
So this process actually happens in real time, well, semi-real time. It's maybe a couple milliseconds, right? It's basically real time, as fast as we can perceive. As the camera is feeding the information to our computer, we're running that localization process, and we have a lot of acceleration using the graphics card on your computer to make sure that that process is really seamless and not slowing anything down. So that localization process is actually happening in real time on your machine. While the acquisition is happening. And so what you finish with at the end of any acquisition, either in MOS, which is our kind of standard software, or in our new tools like AutoAV, is your raw camera images as well as the localization list that's processed in real time.
Fabulous. So we are, of course, interested today in actually what software tools can ONI offer our community to help them make this process more smooth and more simple. So can you comment a little bit on our software solution for analysis?
Sure. I've already kind of alluded to a few of our different software tools here. CODI is our analysis platform, not to be confused with Cori. CODI and Cori are very similar, but it's our collaborative discovery platform. It's actually a cloud-based software that we use for storing and analyzing single molecule data. So in this process that you see here, the localization process is happening locally on your machine, and then you can upload that data to CODI where you can run your analysis. Before showing and maybe like talking a little bit more about those tools, I think it's important to kind of level set what all the different steps of the analysis can be, so you can understand what CODI is and how powerful it is. So if we can go to the next slide. When you're doing your single molecule analysis, in general, it's a sequence of discrete analysis steps, right? I'll go through them in detail in a little bit, but first one is correcting drift. We're acquiring data for thousands of frames. It can take several minutes to acquire one image, and there's all sorts of reasons why there might be some sort of drift on the nanometer scale, right? That we need to be able to correct for. And so that's the first step of any of your single molecule data analysis. Then we'll go into the filtering of the localizations. That's what we were just talking about, right? Making sure that we're only retaining the best localizations that'll give us the best data at the end. Then there's a whole bunch of other optional things. You can group different localizations together if they've been on for subsequent frames. You can, check for a certain area. If you only want to analyze a certain area of your cell or one part of your image, you can obviously set a region of interest to be able to annotate there. Those are kind of all the basic, I would probably want to do those all the time functions. Then we have things that are a little bit more advanced that use those localizations to do more grouping, like clustering, as I mentioned. And if you're doing clustering, you're probably going to want to filter on those cluster properties as well. So there's cluster filtering. And then you want to be able to compare across different channels, imaging channels. So like if for different proteins, the EV example is pretty easy, right? On your EV, if you've got an EV and you've labeled it with 3 different proteins using our EV profiler kit, right, the tetraspanin kit, for example, you would want to see what the different tetraspanin content is for each individual EV. So you'd want to be able to count the different number of localizations across all the channels. So that's what the counting tool would do. And then the next step is you may want to also understand what is the spatial organization now of these clusters, right? In the same way that you've now organized your localizations into clusters, you might want to understand what is the organization of those clusters themselves. And so you might want to calculate the distance between the clusters, or the co-localization, the overlap between the clusters. And so there's so many different things that you might want to do. And each one of those has its own specific settings and parameters and potentially different algorithms that you would need to set in. So it can be kind of overwhelming to get started with single molecule analysis because there's just so many different steps. In CODI, which is, as I mentioned, our cloud-based analysis platform, what we've done is we've taken all of these different bits and pieces and we've turned them into individual workflows that are a sequence of steps that are all linked together, so that you don't need to worry about which individual pieces do I need for my analysis. If you know that you're running an EV analysis, we piece together all the different necessary steps into a linear workflow that you can, with some great default settings, that you can just hit play and it'll run the analysis with those settings. And you don't have to worry about configuring everything for each of the individual tools. So if you go to the next slide, you'll see what that looks like. It ingests, or it takes in the localization point cloud, which is uploaded into CODI in the cloud. It runs this linear sequence of events. You can always go back and change. It's really great. I'll show you in a second. And then what your output is that cluster list, or actually in some of the different tools like our EV profiling tool, you get a summary report, which gives you actually like tangible information about what is the different phenotype information for each of your EVs.
Fantastic. And just to confirm, because I had a lot of anxiety in the past analyzing my localization data, because I didn't have this tool at that moment. Just to confirm, the user does not need any coding experience to analyze the data. Is that correct?
No, yeah, totally. No coding experience necessary here. We've, as I mentioned, kind of wrapped up all those tools into individual widgets, is what we call them, individual steps along the way. And we've exposed some of the parameters that are required or nice to kind of be able to tweak, including sometimes different algorithms. So like drift correction, for example, there's a few different algorithms that you can use to correct the drift. We have defaults that work well for certain sample types, but you might want to be able to tweak what those look like. So it's a good balance between, one click and just get everything done, and also still have the configuration that you need without needing to understand the code behind the scenes.
That sounds fantastic. And another question that is very popular normally for the users is regarding the drift correction. So what do I need to perform a good drift correction? Do I need any help from fiducial markers, or can the software handle it on its own?
I would like to show that to you, but in general, what we have in CODI, and what's a kind of standard practice, is what we call cross-correlation tools. And so what these cross-correlation tools are doing is they're aggregating a bunch of different frames, so like let's say 100 different frames, making it discrete sequences, and then looking what the overall drift is like across, very simply, across the entire acquisition, and then realigning all that over time. And then same thing for multiple channels and making sure everything is aligned. It's actually quite a bit more complicated than how I'm explaining it. If you have a sufficient number of localizations in each one of your image, those cross-correlation algorithms work really, really well. But you have to have a relatively high number of localizations in your image. So if, again, I'm using the EV example here, but if you have an EV sample that has just really low, or just like very low number of EVs on your slide, or your density of labeling for that specific protein is very low, you might want to introduce some fiducial markers like fluorescent beads to ensure that you have a high number of localizations in each frame so that those algorithms can actually work.
Perfect. So I see our user base, our audience today, they are already ready to use CODI. So we have a question from the audience saying, how can I actually get a CODI account? How can I get started?
Oh, cool. I actually didn't Well, maybe we can drop a link in the chat here. CODI is open to everybody. You can access it on alto.codi.bio. Maybe our organizers can drop a link in the chat here. You can sign up for free, and actually, maybe I can show you what it looks like once you've signed up. You can use your email account, or you can, if you've got a Google account, you can just sign in with Google. It's really fast and easy. And once you've signed in, you can actually, you don't need to have any data of your own, any ONI data, to be able to start using CODI, because there's a lot of publicly accessible data sets that our users have shared publicly intentionally, so that anybody can access their images, run some analysis on it, take some screenshots, like do all the things that you'd want to do with your data, but they've shared it publicly. So you can actually get a feel for what it's like to use CODI and have CODI for free.
That sounds amazing. Please do try it out and let us know how it goes. But I think we arrived at a moment that people were very curious for. Corey, can you do us a big favor and show us how this CODI platform actually looks like and works, and maybe an example of how you would interact with one of the pre-made workflows?
Yes, I can. Let me just make sure that I'm sharing correctly. Do you guys see my screen now?
We see the welcome page of the CODI.
Welcome
Yes.
to CODI. Perfect. So when you guys sign up for CODI, and I implore you to do that right now, it takes just a few minutes to go sign up for CODI, and the link that I just saw drop in the chat here. This is the first thing that you'll see, is a list of data sets, and you're like, where do these come from? They're publicly shared data sets that all of these different users have shared intentionally publicly for anybody to use, right? So you can scroll through and you can see here, if you want to, you can share your own data on here. Otherwise, you can just see all the other beautiful images that people have shared publicly, which is really great. What this means is that you can use CODI and start fooling around with all the different tools and get a feel for what it's like without having to actually have any data of your own to try it on. You'll see at the top here, I've got a bunch of different tabs open because I've kind of selected some data sets that I want to show you. But obviously, just like very general CODI walkthrough, again, this is in the cloud, right? This is a website that you're going to. You can upload your data here. We actually have a desktop tool as well to facilitate the upload from your machine. Or if you're using one of our new tools like AutoEV, the upload happens automatically during the acquisition process. So you don't even need to worry about that. It just appears in your CODI account. It's great. You have your own private data sets. You have the ability to share data sets with other colleagues or anybody else, really. So you can share it with any individual that's on CODI. It makes it really easy to share data sets with, as I mentioned, with colleagues. And the real advantage there isn't just sharing the data sets, but you're also sharing your analysis. And so you can one person can acquire data, and everyone can kind of analyze it in their own way and find what are the best settings and whatever, and share it across the teams. It's actually really cool. We've got collaborations with just folders, basically, for storing all your different data sets. And then we have some acquisition apps like the AutoEV tool that I was talking about that live in here. So let me just show you this example.
We see a little bit cut off on the analysis pipeline. If you can just squeeze your window a bit more.
Like that?
Perfect. Amazing.
Okay, interesting. Okay, learning about the GoToWebinar. Okay, so this is a data set that I have here. If I go back to the dataset info page, it's a human T-cell derived EV. There's 3 different stains on this one. I think this is from our EV Profiler 1 kit. One of the cool things about CODI is that in addition to obviously storing the data set, you can save these custom views. So if you find an EV that you really like, or you used in an image in a publication, you can save that specific view. So that zoom in with all the features that you have in your CODI analysis, you can actually save that as a view to get back to later. And that's what you see here. You'll obviously have all the details of like the acquisition information, which may or may not be relevant for what you're trying to do. But then more importantly, you have a list of all the analyses that have been run on this specific data set by myself. Actually, I don't even show up in this list because I haven't run an analysis on this recently. I do see Laura's name on here recently from a couple of weeks ago, as well as any user, because this is a publicly shared data set. So anyone can come in here and run some analysis on it. To run an analysis, we actually, like I said, have these really great workflows. So if I wanted to start from scratch, I would click at the Add a New Analysis tab, and you would select from one of our different analysis workflows that we have. This is EVs, so I would choose EV profiling. And then from that list, I would choose what is the right settings for my specific acquisition. This was an EV Profiler 1, I believe, setting. So I would typically choose EV Profiler Essentials because that was from our original kit, but we also have settings for the EV Profiler 2 kit that are for all the different variants of the 3-color tetraspanins, the ones with the pan-EVs, with a custom biomarker, without a custom biomarker. So these are starting points for your analysis settings that are very specific to the kits that you have. And hopefully you'll get an understanding for, okay, this is the right default settings for me to start with, depending on what my sample is. You'll also see that I have this really cool one, my settings with the cool little geek face that I've saved myself here as a user. So anytime that you create some custom settings, so like you'll run through maybe with somebody like Laura, right? The first time that you're setting up or you're running a demo, or the first time you're setting up your machine, you'll go through and you'll tweak some of those settings that I'll show in a second. You can save all those and then reapply them later, which is really cool. All right, so I just want to get back to the exact analysis result that we had here before, which is this one here. All your analyses show up as little tabs on the left. So you can run multiple analyses on the same data set at the same time if you want to, or you can really quickly compare different analysis results. The first step of all of them, as I mentioned before, is drift correction, right? So the question around what it is and how important is it, I can show with this data set, because you can see with our graph here, it's detected over 1 micron of drift in this particular sample over the course of the acquisition. So you wouldn't notice because we've applied the drift correction here and our EVs look really great. They look really round and they're co-localizing across the 3 different channels. But in CODI here, we can actually turn off the drift correction visualization. And you can see what it looks like. It's a big mess, right? This is, I'm not sure that any clustering tool would pick this up effectively. I don't know that anybody would look at this and say this is an EV. It looks like a line to me, right?
This is worst case scenario.
Worst case scenario. And I can talk a little bit about that. I mean, if there are specific questions about how to address this, just in general, this drift correction on our machines would generally come from changes in temperature, right? And so what might have happened here, I don't know the details of this acquisition, but what's probably happening is that the system was either probably heating up while this acquisition was happening, and pretty drastically. It might have been in the winter in our Oxford office where it's quite cold, and then they turn on the nanometer to start heating to 32 degrees, and then you can see this kind of drift over time. And so the one recommendation is make sure that your system is stable in temperature before you start any long-term acquisition. Doesn't necessarily need to be at 32 degrees, but just that the temperature is stable for the imaging that you're doing. And again, our tools like AutoEV give you warnings to make sure that.
We have one question, sorry to interrupt, because it's not only temperature, but Miriam from the audience is asking, what about long acquisition times? How precise is it to still correct this data? Will that also work?
Yeah, I don't know that I have an example here, but this is a pretty short acquisition, right? 5,000 frames, but you could imagine acquiring 50,000 frames, and the stability of the algorithms, let's say of the drift correction, as I mentioned before, really just depend on the number of localizations that you have in each frame. So as long as you're continuing to do localizations where you've got, a number of, 20, 30, 40 localizations in your frames, your drift correction algorithms will still work well. Where that will fall down is if you're trying to, really image every last fluorophore of your sample for some reason, and you're trying to go really to the end of the sample life in terms of blinking, and you're reducing down to just a few localizations per frame, then maybe your drift correction might start deviating. But at that point, I'll show you some tools in CODI here to restrict the number of frames that you're looking at to make sure that you're not including anything that might have not drift corrected correctly. One thing you can really look at in our graphs here, it's important, is you can see the quality of the drift correction with the curves. In general, you'll have these kind of linear trends, right? Because in general, this drift correction is thermal, and that thermal expansion is a pretty linear process. You might see little jumps, and those are generally when we're changing channels of the imaging channels, right? So when we're swapping between the 640 channel and the 560 channel, the 488 channel, and that's just the drift correction algorithm centering the different channels to make sure that they're spatially overlapping.
And also, how about, you mentioned already to figure some settings, but how would I go ahead, is the audience asking, manually adjusting some of the drift correction parameters, if you can just
Totally. It's a good question, because I mentioned that we do try to have this balance of, not needing to code, but also still have some level of tweaking while retaining the ability to just hit one button to run the entire workflow, right? So we have on most of our tools this three-dot menu in the top right. That we call this the back of the widget, and this will expose some of the settings. And so there's a few different settings that we can do. We can change the algorithm, and that's what you see here. We've got the drift to minimum entropy, we've got a cross correlation, two different cross correlation methods. And all you have to do is click on this button to be able to switch that, and I can show you what that looks like. So when I do this, what it's going to do, it's going to branch a new analysis. What that means in CODI terms is it's taking whatever previous analysis you had, it's remembering the bits that came before. In this case, there aren't any pieces before drift correction, so it's starting over from scratch. And it's allowing you to create a new analysis without destroying the old one. So this is like a totally non-destructive process where you're retaining your old analysis, and now you can try out some new parameters. So if I wanted to see how the direct cross correlation method compared, and if I wanted to manually set the number of windows that is used for the cross correlation. So in that example of like, you have a really long acquisition, maybe you don't need to have sequences. You can have your sequences of localizations that are being linked together. You can have them be shorter or longer or whatever. You can change that. You can actually modify the number of windows, and the calculation there is just 2 windows would be, if your entire acquisition is 10,000 frames, 2 windows is 2 chunks of 5,000 frames. It's probably not the best way to start. I would do something more like 10 or something like that. If I can enter a number, let's see if, there we go.
So even though we have the basic recommendation, standard setting, it is still possible to have a look and maybe in challenging examples such as this one to manually adjust my drift.
That's a really great summary. So I'm just trying some random values here, which is honestly, like, sometimes when you're getting started, that's what you got to do, right? Just figure out what this tool does on your sample. And then that kind of leads me to the next point, which is you can save those settings. If you find it works really well, you can save that just for that tool by typing in a name here, and that'll show up and you can reload it from the load settings here. Or when you're done with the entire workflow, you can just hit run all steps, and that will turn into and see, you can see it's kind of piecing back together here. That actually worked really well, right? You can see it applying the drift correction that we just created manually. I can come back here and we have a much more linear curve. So maybe we just manually found a really nice drift correction for this particular data set. I'm gonna hop back into the old analysis that we had before. Go ahead. Sorry, Laura.
Because there's a lot of interest in the drift correction, so I'm going to go ahead and ask more questions regarding that.
Yeah, let's do it.
I have a question regarding how does this differentiate between actual biological movement and drift, especially in high dynamic samples?
That's a very good question. The drift correction is basically assuming that you're using a fixed sample, right? So for storm analysis, and that's primarily what these workflows are for, it's assuming that your sample is fixed and that all that's changing is the blinking of the fluorophores on a fixed sample. If you have a dynamic sample that you're trying to analyze here, I would suggest using fiducial markers like fluorescent beads to have these kind of static localizations across the field of view, so that these cross correlation algorithms can latch onto those, and that's what it would be using for the drift correction. So here I'm just showing how we can easily compare across the previous settings that we had, the default settings, versus the ones that we just manually had. You can just easily swap between 2 analysis tabs like this to see the different results. So you're staying, in the same spatial location in your image, but you're just seeing the differences in the results of drift correction. You can see how important drift correction is as a first step in getting it right for the rest of the analysis.
All right, so once we have figured out our drift correction and we are happy, what is our next step?
The next step we would do is localization filtering. What I'm going to go ahead and do is have it run all the next steps subsequently in this list, just to kind of get ahead on the analysis. That shows you how easy it can be to run your analysis in CODI. Once you've got your analysis settings set for each of the different widgets, either using the defaults or the ones, like I said, that you'll probably go through the first time and set each one manually, then you can just hit play and apply it across all of the different tools. So you don't have to click 20 times for every data set. And this also allows us to apply those settings to a lot of different data sets all at once in something that we call batch, which I'll show in a second. So the next step would be to do the localization filtering, which is the step that we talked about a lot in the intro here. CODI has some really great tools for doing this. The best one, let's see, that I can show probably is maybe the Sigma. So the sigma is the size of the point spread function. If you do the Gaussian fit of the point spread function, which is what we're doing, sigma is that 1 over e value, I believe, is the sigma value. Anyway, it's a metric that gives you roughly an indication of the size of the Gaussian function that's estimating the size of our point spread function. And so what we would want to do is make sure that we're setting our sigmas, looking at the data, which is what we see here in the 3 different channels, and we can maybe just look at one channel to make it easier, like that one. What we want to make sure that we're not seeing is like 2 peaks in our data, where we have a really large small peak and then another larger peak. Because if we have a larger peak around twice the size of the first one, that's probably some stuff that is 2 localizations on at the same time, and it's this large point spread function. And so we'll set our sigma values, our sigma filters, to make sure that we're roughly around 250 maximum size, so that we're not localizing anything or capturing, in the rest of the analysis, any localizations that are larger than they should be, which is, for those single emitters. In the same way, you could do the same thing with photon count. Let me just, oops, sorry. There we go. It's a little hard to see the graphs here sometimes, so you can actually change the axes options. So let's go to X, and then, because it'll auto scale to like the largest value, which is sometimes really, really high, especially for photon count. Just change these a little bit here. 5,000 maybe. Okay, so now you can see the graphs a little bit better. This data looks like it was acquired really well in very good single molecule conditions. Otherwise, like I said, in your histograms here, you would see probably multiple peaks, or you would see something that doesn't look like this very clean distribution. You would see something like a long tail or something like that. And at that point, we might want to modify our photon count filters to make sure that we're not getting things that are too bright, because we wouldn't expect a single fluorophore to be that bright, right? It's probably a group of fluorophores that's not quite blinking or something like that. So these are the kind of settings that as you can see, we had some great defaults in here because we understand our systems really well. We know the EV profile, we know what to expect here. But as a user, you can obviously come in here and modify all these things. Same thing with localization precision. You can look at the ones that are only the most precise if you want to, or only look at the ones that are the least precise, or whatever you want to do.
So Miriam is asking, what are the best practices for validating the accuracy of molecule counts obtained from CODI? So what would you advise for that?
For molecule counts, let me make sure that I understand the question correctly. So part of this process, right, is we're localizing pretty much everything, and then we're doing the drift correction, and now we're doing these localization filters to make sure that what we're running through the rest of the analysis is just our true single molecules that are coming from the images that we had. So it's a lossy process there, right? We are intentionally excluding any data that may have been acquired that we're deeming imperfect in some way to our localization process, right? So it could be like the typical example, 2 emitters on at the same time, or some background fluorescence that's somehow being localized, something like that. Part of that process means, and because of just like the stochastic nature of the blinking to begin with, it means it's quite difficult to get like true molecular counts as a quantification from storm images, for example. But what I would say is, statistically, if you're applying these same kinds of filters, right, if you're acquiring your data in a very, very good single molecule regime, and what that means is your blinking buffer is in, really fresh, your acquisition powers are set appropriately so that they're blinking, the molecules are blinking well, but not bleaching too quickly, and all of that stuff, right, all of those steps we can work through with you guys. If you're doing that, then if the input data from that camera that I showed at first, right, the blinking data, is really, really good single molecule data, you're not going to lose a ton of stuff, a ton of information through the localization filters here. So I don't think I answered your question directly, but basically I would say if you want to make sure that you're doing it well, make sure that the acquisition has the best single molecule data as possible so that your analysis can be as faithful to those acquisitions as possible.
Best signal to noise, as we like to say.
You got it.
I think people are now very curious to go into the clustering part. Can you show us a little bit about the clustering? And maybe on the side, if you can just comment a question from the audience regarding how can we distinguish using clustering what is a biologically relevant cluster and what is actually an artifact?
Artifacts are hard, but there's some that we can identify in a very systematic way to help you understand in the EV example. Sorry, it's what's in front of us right now, but like what is an EV versus what might be just background fluorescence from an antibody that's somehow stuck to the cover slip, but isn't attached to an EV, right? Clustering is actually a really great tool for that. So let me just get into clustering first, then I'll talk about how we can do that. So what clustering is doing, as you may have just noticed, is it's looking at all of these localizations that we have in these EVs. It's actually more helpful if we zoom out a little bit. So we've got all these EVs across the field of view. And when we run our clustering algorithms, it's finding spatially, those spatially distinct groups of localizations and clustering them into one object, right? So each one of these different colors is a different EV, for example. And you can click on it and you can see how many localizations are in there and roughly the size by area. So that's like a convex hull calculation. You can see here as well a distribution of the number of localizations to help you understand roughly how many localizations are in each of your clusters over time. This particular algorithm is called the HDB scan algorithm. It's one of many different ways to cluster your data. For EVs, it's the algorithm that we found to be the most robust and specific for EVs that gives you the best results. There's only one real setting that you have to set, and that's what we call the EPS radius. It's a parameter of the algorithm. Roughly, I know this is like a little strange, but it's roughly the size of the EVs that you're expecting. So between 70 and 100 is probably a good starting point. And you can see when you do that, that we're able to distinguish, let me see, it might not be super visible on the screen here, but like we're able to distinguish this cluster in red versus some like kind of background spots that are targeted as noise. Let me just turn up the brightness here a little bit so you guys can see. So these dots in yellow, when I run the localization, here I'm turning on our noise, so showing what is not part of the clustering if they get blacked out. So those specific things that are kind of, it may have been a fluorophore floating in the background during that acquisition. We're not entirely sure, but it doesn't look like it's an EV, right? And so what our clustering is allowing us to do is to say, hey, this is an EV, this is not, right? And so it becomes noise. There's a whole bunch of other settings that you can set in the back here if you really need to tweak that. But in a first level, just doing the clustering step itself will filter your biological structures like your EVs from background. The next step is cluster filtering. And so if you're finding that you're still getting a lot of clusters from localizations of individual fluorophores all in one space, and you can't separate them, like your clustering parameters are setting it up so like your clusters are also giving you some background signal, you can use the cluster filtering step, which is just like the localization filtering step, but now we're applying it to our clustering data to really narrow in on the kind of clusters that you're looking at. And again, we have some defaults here, and the noticeable one is the radius of gyration, which roughly is the radius of your cluster, even though your cluster is not always circular, right? So just roughly the size, but the radius is like half size of your cluster. And you can see here we've set it to 20. And so by default, what that means is that we're going to remove anything that's smaller than roughly 40 nanometers in diameter, which is roughly going to be the size of like a single fluorophore stuck to your cover slip, right? We don't expect EVs to be that small, at least for this particular sample, and so we're okay filtering out the things that are smaller than that. And so what that means for our subsequent analysis is that we're not going to be integrating anything that was maybe noise from whatever mechanism, right, biological or other, into our subsequent analyses, and just keeping only the EV data for these things that are, roughly circular, roughly this size, and have a good localization density.
And how about, can you comment? This sample looks very nice, very, very dispersed. How does CODI handle cluster overlap in densely packed samples? Can you comment on that, and what would be your advice here?
So yeah, dense is hard in general, right? It's especially for clustering. And so there's a few things that you can do. You can play around with the EPS radius here. We have some different tools here for minimum size and minimum number of localizations that would be part of your clusters. But if you're finding that these tools are not sufficient, we actually have a whole bunch of other analysis workflows that include other kind of clustering algorithms that give you a lot more tweaking that allow you to kind of cluster what you want. And so I think I have an example in one of these tabs here. Let's see. So this is a different workflow. This is our clustering and counting workflow, which is the one that gives you the most options, let's say, right? The most flexibility. The EV one is probably the most narrow because it's very specific to our EV application. This is the one that if you're doing just generic cellular imaging is probably the one that you'll want to start using. And so the clustering tool in this particular workflow has a few different algorithm options. So you're able to use the dbScan like we had in EVs, but you're also able to use our constrained clustering algorithm. And this is really, really, really powerful because it allows you to constrain, instead of what we did before, which is we do the clustering and then we filter out a bunch of things. What we can do with this algorithm is tell it kind of what we're looking for, right? So if we know that it's very dense, mitochondria, let me show you what this looked like before the clustering. These are mitochondria, right? Let me turn off this channel here and maybe make it grayscale, might be easier for you guys to see. These are, maybe the overall it's not super dense, but the individual clusters are pretty dense, right? And so you've really got to tweak your settings to make sure that what you're grouping together, if what you're interested in analyzing is as a cluster is an individual mitochondria or a group of mitochondria, you really have to play around with your different parameters here. And so you can in real time see how changing the filter, it's not filters at this point, it's inputs into the algorithm. So it's what is the smallest number of localizations that I'm looking for in a cluster. You can kind of tweak that in real time and see what the impact is on your clustering results as you're fooling around with it. So if you want things that are a little bit smaller, a little bit larger, you can kind of play with multi-parametric inputs into these algorithms to get the kind of results that you're looking for in denser samples. Takes a little bit of time to find the right settings, right? But what's really cool is the interface is really reactive, right? So you can do it in real time and figure out what are the right settings and then move on to the next step.
So I see in this algorithm, we don't have the EPS parameters such as in the previous one. So would this be easier for samples that I maybe don't know exactly the size range yet?
Yeah, I think the dbScan algorithm works really well when you kind of have an idea roughly of the biological structure that you're looking at. If you're starting from scratch and you don't really know, I would suggest you start with this workflow, the clustering and counting workflow, and use our constrained clustering to first just kind of play with it. And like I said, CODI is free for all of you guys to try out. You can just go to the alto.codi.bio. This data set is a public data set that's there. If you want, we can share it in the link in the chat or with you guys afterwards if you want to be able to analyze this specific data set and fool around like I'm doing right now with these different filters to see what it feels like.
Pramesh from the audience already registered and gave us a feedback that it was very easy for him to join, and many thanks.
Yeah.
Great to meet you, Pramesh. Please explore.
Other cool thing, so because it's a website, sorry, cutting you off here, but like you can just right click and say copy link address on any of these tabs, any of these analysis results, and you can share that directly with anybody. So that's what I'm saying. If you wanted to come here, you don't need to search for this data set. We can share that with you. You can share with colleagues. It's great.
Wonderful. So I think we covered a big part of the clustering and how we can form the clusters. And what is next? What do I need to do? How do I get my data out?
That's what's next, right? So there's a few different ways. I'm coming back to our EV data set here. There's a few different ways to get your data. In the last tool of any of the workflows, you'll have a little download button. So here, this is the summary report, which is, as I mentioned very briefly earlier, in our EV analysis workflow, gives you an output for the positivity. So that's what we call basically the phenotyping of like, is it single positive, double positive, or triple positive of your EVs. And so you can just click this like download button and it'll download those CSV results so that you can plot them in another program like Excel or Origin or Prism or anything like that. That's for a single data set, right? And that's not hugely relevant for a lot of studies. I think what's more important in general for our users is to be able to then take these settings that you've now tweaked and apply it to all the data that you've generated during your acquisition. So like I like using the AutoEV example a lot. In AutoEV, we're going to by default, acquire somewhere around 16 fields of view for your experiment, right? Across the 4 different lanes, you're going to have a lot of data. By default, you'll have some analysis settings that'll be running on there. But if you want to then apply this tweaking that we just did with all those clustering settings to all of your different data sets, you can hit this next step button. You can save the analysis results if you want. I can say this is like our webinar settings, right? So that I know for later I can do it. And more importantly, you can click this use settings and batch analysis to actually apply this to a bunch of different data sets. And so while this one doesn't probably have the best chances here, because these are all different kinds of data sets in this collaboration. Let me show you what it would look like if I'm on an example here where this is all EV data that's been acquired to yeah, in this kind of single acquisition. So I can go to my analysis results here. Here I can see all the different analysis results for all of the different data sets that are in that collaboration, and I can say batch download, right? So once I've applied it to all those different, actually, let me show you how to do it a little bit different. That's the next step. That's downloading the data. First, you want to start the batch analysis. So that's starting the batch analysis here. You would do EV profiling because that's what we have. And we can either do my settings, or if I want to see my demo, my webinar settings, I can click show all because I didn't favorite it, right? So I can apply that here if I want to. And then I just select the data sets I want to run it on and hit run. Super easy. This is all in the cloud. It's not going to slow down your computer. You can actually close this window, open it. You can do this on the machine where you acquired the data. You can go to your desk machine and then watch it processing, or maybe by the time you got there, it's already done. Right? That's one of the best things about CODI is it's not locally on that machine. It's really anywhere. You can, I've done this before. Start the analysis when I'm leaving work, and by the time I'm home, it's ready for me to download the results so that I can start doing some post analysis afterwards. So that's where I was getting at before. Is then you would want to batch download your results from those, and so I can just select a few here that are completed, just for example. And then I can't get rid of this little guy here for some reason. Get rid of the download window. But anyway, there's a download button. Let me make my window a little larger. Okay, probably get it there. Download results. And it would download a CSV that has, it's actually combined all of the data from all of those different acquisitions into single files, so single positivity reports, single clustering files, so you don't have to aggregate all that data yourself. One of the questions we had earlier, right? Do you have to be an expert? You don't even need to know how to aggregate your data or spend too much time in Excel aggregating it all, because it's all already there in a single file for you to analyze in some other software using or plot graphs with or anything like that.
Wonderful, because some of the questions also we have a lot in trainings is like how accessible is this data that I'm generating? Can I do something with it? And indeed, just to emphasize, you will get the CSV file that you can open with any computation program such as Excel or whatever your program of choice will.
The nice, maybe one next step, I don't know if we want to hop back to the slides, but I dropped an image in there of one of the things, if you're using AutoEV, which is our kind of end-to-end acquisition app, where it's going to acquire your data, it's going to analyze it in real time. It's also going to provide you a report that gives you some really interesting and important feedback while you're still at the microscope, while your slide is still on the machine of, should I continue to image? Do I need to acquire more fields of view? Does my data look like how it needs to? Do I need to tomorrow redo this entire experiment because my sample isn't prepared correctly? There's a million reasons why you want that information very quickly. And so we've prepared this really great single page report that happens, that shows up in AutoEV when you're done acquiring your data. You can also regenerate it now from CODI. Once you've acquired data with AutoEV, you can regenerate that report inside CODI. I don't know that I have any example data here to show, but maybe we can hop back to the slides and just really briefly show what we're able to do today with AutoEV and kind of the ethos of CODI going forward, where we'll have a lot more integration between the acquisition, the analysis, and then these kind of reports to make the data outputs a lot more tangible and understandable for anybody in front of their microscope.
Fantastic. We had that question yesterday, I have to say, that one of my users said, I want a really simple solution. I want to have a really quick look. How do I generate graphs quickly? And indeed, one of the solutions is our new software, AutoEV, we provide. So if we can just have a quick peek at how does it look like, this AutoEV report, then we can go through the characteristics that it shows. We will need to get back to the main deck of the slides.
Yeah, looks like it's swapping over here.
Yes, some technical delay, no problem. And here we have an example again with extracellular vesicle analysis. Corey, can you walk us through this report?
Totally. It was really great to design. We talked to a lot of customers who use our UV profiler and asked them, what are the key metrics that you're looking for when you are done with your experiment? What are the typical analyses that you do with all the CSV files that we said that we spit out from our standard EV reporting? which the word report can mean a CSV, which is what we were talking about before, or this kind of like one-page PDF report, which is more the general term for reporting. So there's 3 key metrics that you can see here and really easily compare across the 4 different lanes of your EV profiler chip. The first one at the top there is the number of EVs per field of view. So here we've acquired 12 total fields of view, 3 per lane. And with this, just seeing the number lets you evaluate, okay, do I have a pretty homogeneous, distribution of EVs on my chip? And also, how many EVs do I have do I have enough statistical power in my analysis here to do the analysis that I want to do afterwards, right? You might only see 200 EVs per field of view, and I'm going to need to go back and acquire some more fields of view to be able to have enough statistics for these particular kinds of EVs that I'm imaging. And you can obviously compare across all the different lanes here. So you can say if you have a control sample in one lane and you have a patient sample in another, or a therapeutic sample in a different one, you can compare really easily across the conditions. The second information that we have here is that positivity, the phenotyping, or the other way of saying it is like the individual, single, double, triple positive. And the color code here matches what you saw in CODI, right? So the blue, the cyan, the magenta, it's the same colors that you were using to acquire those different proteins that you saw in your analysis. And that's what's showing up in the report here as well. So mentally mapping those things is really easy once you start getting into it, and you can really easily compare the different positivity information across the lane. So you can see if one sample, if you're expecting, our new EV Profiler toolkit has a permeabilization region in it, right? So you can do a permeabilization and non-permeabilization test, and you should really quickly see that when it's permeabilized, you have some double positives for the protein that's inside versus the one that's not permeabilized. You wouldn't expect to see that double positive population. And then lastly, we have size information, and the size information is shown as a general distribution, that little histogram there on the grid on the left, but then you have per phenotype or per positivity class size distributions. And so this is really, really cool, because you can say my triple positive ones look like they're a little bit larger than my double positive ones, or maybe it's the inverse, or the ones that are positive for this particular biomarker tend to look like they're a little bit larger than ones that are larger or positive for a different biomarker. So you get all that information in one report while you're still in front of the microscope, or you can generate it afterwards. So it can really help you understand, well, do I need to continue imaging? Do I need to change my sample? Do I need to change my labeling? Like, there's so many different things that you can understand from this one sample.
Can you maybe quickly comment on how do I assess biomarker heterogeneity in this report? In what part see that?
There's 2 places really. I would really focus in the center in that bar chart. When we're talking about biomarker heterogeneity in general, we're going to talk about the different positivity classes, right? So what you might want to be looking at is if here was, for example, patient samples, right? Maybe you have a couple different patient samples that you're running the same biomarker panel on, right? 3 different proteins that you're trying to see. Are these biomarkers present in a healthy control versus some diseased patients? With this graph, you would just look at the different colors. The different colors represent the different positivity classes, and you can really easily compare against those different conditions who has, like, which of those conditions has more or less of the specific biomarkers that you're looking for. And by having it all outlined together horizontally, it makes that understanding really, really easy. That's why it's laid out this way. And then next level, sorry, I'm getting ahead of myself here, but is more heterogeneity could come from the size populations, right? And that's also why it's laid out in this way. So in those, you have the information about the relative percentage with the bar chart of those populations, and then you have the size information below to compare. So you can see in one condition, that population is larger or smaller than another.
Absolutely. And I feel like we could talk for hours about SMLM analysis, but our time
Too easy for me.
Almost up. So I have one final question. I hope you can answer it very briefly, and it's regarding what's in the future. Will AI help us to do analysis faster and more accurate? So what is your take on that?
I think so. I think for sure. I think single molecule data at the image level and also at the just point cloud level lends itself really, really well to machine learning algorithms and AI in general. And we've actually piloted an AI EV profiling tool that's trained on our EV data. If you're an EV profiler to an auto EV user, you get access to this tool. I don't have time to show it today, but what it does is we saw all those different parameters that we had to put in, right? All the tweaking of the EPS radius and the filters and all those things to make sure that what we're looking at is individual EVs. we've trained models that understand what is an EV and what isn't, and the AI tool doesn't have any of those settings. You just hit play and you get a result from your clustering that is just your EVs, because we've trained those models to understand what are EVs and what isn't. So I think that's just a kind of an inkling of going forward what the machine learning and AI tools can do for single molecule analysis.
Thank you so much, Corey. And thank you for joining us today. I hope you had a good impression of how our usual single molecule localization microscopy analysis workflow looks like, and specifically how CODI looks like. And just as a reminder, it's an open platform. If you go to alto.codi.bio, you will be able to access it. And that already completes our session. I hope you have learned a lot. Thank you for joining this session, and thank you for joining the previous session if you were also on that one. And I hope this was very useful for you guys. And yeah, I want to thank Corey for today. And stay tuned for future webinars. We will continue to do webinars in the future as we will continue to develop new tools for you. So just stay tuned on our website, oni.bio, for the latest updates that we can offer you guys. So thank you very much.
Thank you, Laura. Bye, everyone.
Bye, everyone.